Well, good evening, everyone, and welcome to the Brian Crombie Radio Hour on Saga 960 AM. I've been extremely interested, as probably all of us have been, in COVID-19 and some of the different people that are working on vaccines and on therapies and on PPE, et cetera. And so last week we interviewed a group at the University of Waterloo that are developing a vaccine, and we've had a couple different people talking about clinical trials. I've now got a group of people that are with an institute that's associated with the University of Saskatchewan. It's on the University of Saskatchewan campus, but they're a separate organization, and I want to introduce you to them tonight. We've got Darryl Falzarono, we've got Volker Gerdts, and we've got Paul Hodgson. Gentlemen, how are you all?
Good, how are you? Thanks for having us.
My pleasure. So tell us, what have you got?
So maybe I'll start. So we have a vaccine. We started to make this early January. The vaccine is already being generated. It's been tested now in animal models. So really, VIDO INTER Vac, our organization, and Darryl in particular, we were the first in Canada to isolate the COVID-19 virus. We were the first in Canada to establish an animal model for it. And we're now the first in Canada to test a vaccine or have a vaccine candidate in animal testing.
Okay, let me interrupt you just for a second. Ferrets. I understand you're doing it in ferrets. Why ferrets?
So they are a very good animal model for human respiratory viral infections, influenza and others. There is some receptor homologies. The receptor that is being used by the virus to get access to the cell is very similar between ferrets and humans, and so that's why they're one of the animal models that are being used. Okay. Another model is the hamster model, which we also have established here, and then of course people are using non-human primates too.
Excellent. Sorry for interrupting. Please go on.
So then the important thing is that we now have already started to manufacture the vaccine, so the preclinical material. And that will allow us then to do safety studies, which are paramount to all of this over the summer. And then we will start our clinical studies in the fall of this year.
Excellent. And as you mentioned, you worked on SARS before. I understand you also worked on MERS. How does the time frame, the timeline that you were working then and now compare?
So everything, of course, is much more rushed, as you can imagine. Our organization worked on many coronaviruses, on porcine, so pig coronaviruses, cattle, and then as you mentioned, also on MERS. And so now everything is much more accelerated, much faster, and much more focused on getting into the clinical phase as quickly as possible.
And you've gotten a fair amount of money from different government grants, is that correct?
That's right, both from the federal government as well as the provincial governments.
Excellent. How do you go about getting that? Is that like an application process, or what is it?
It's a combination of application processes, us reaching out, phone calls, video conferencing, a lot of different mechanisms.
Okay. And tell me about your organization. You said that it's at University of Saskatchewan, but it's separate. Can you explain that?
So we are a research center here at the University of Saskatchewan. We're somewhat independent, although we're part of the university. We have about 160 staff members here at the moment. Some of us have dual appointments as professors on campus as well. Others are research scientists. And our mandate always has been to provide solutions for the industry through our research, and that is both the human health as well as the animal health industry.
And so maybe you could tell me what each one of the three of you is responsible for.
So I'm the director and CEO.
This is Paul speaking. I'm one of the associate directors. I'm responsible for business development, helping with strategy and communications.
Darryl?
And I'm a research scientist, so I am running all of the lab-based studies and involving this and coordinating all that work.
Excellent. And so when do you think you'll have a vaccine?
So we'll have a candidate that will be tested in humans by the end of the summer, late maybe fall. And we, if everything goes well, so the human phase 1 and phase 2 testing will go as we plan, then we hope to have something sometime middle of next year.
And middle of next year would be what, to start commercialization, or would you be ramping up full speed ahead?
So normally what comes after what is called a phase 1, phase 2 trial is then a so-called phase 3 trials, but there is ways under which the regulator can fast track that. So we might be able to provide the vaccine already sometime mid of next year to targeted populations.
We're chatting tonight with three research people from VIDO INTER Vac, associated with the University of Saskatchewan, about their vaccine. We're going to take a break. Stay with us. Well, welcome back to the Brian Crombie Radio Hour. We're chatting tonight with Darryl, Falzarono, Volker Gerdts, and Paul Hodgson, three gentlemen that are part of VIDO INTER Vac, a vaccine company associated with the University of Saskatchewan that have been one of the first people to have a vaccine ready to go into animals. They've got quite an experience on vaccines previously, and it is a vaccine for COVID-19, obviously. And they're quite hopeful that by the middle of next year, you'll have it ready for phase 3, or you'll actually have it ready to start dosing to people in the public?
Hopefully starting to give it to targeted population, people that are in urgent need for it.
Excellent. So tell me, how is your vaccine different? Moderna, this company in the United States, has got a lot of press on how their vaccine is different. I spoke with some people from the University of Waterloo that are using something called a bacterium as a delivery mechanism. What's the secret sauce of your vaccine? Any of you? Hello? Have I lost you?
The virus. I'm still here.
Okay. What makes your vaccine different?
So we're using a proven technology which is called a subunit vaccine. It's just individual proteins, and they're formulated with adjuvants, and these adjuvants are being used to enhance the immune response to the protein.
What is
how about I let Darryl explain a little bit the details around it.
Okay, Darryl, what's an adjuvant, please?
So as Volker was saying, so something that enhances the immune response. So if you give someone a protein all on its own, usually our body would mostly ignore that most of the time, even if it's foreign, or the immune response we would induce would not be sufficient to protect you, for example, from a virus challenge later on, or a virus that you might encounter. So because ours is a protein-based vaccine, which is probably one of the most common and safe routes of doing a vaccine, we need something to increase the immune response to that, and that's what the adjuvant is. And in our case, that's 3 different components that are combined together that are enhanced immunity compared to if you were to use them alone.
Okay, and my simplistic understanding of vaccine is you're actually giving someone a small dose of the actual virus. Is that correct?
So in this case, no. It's a part of the virus. So we actually take about half of the protein that's on the surface of the virus, called the spike protein, and that's what we're using in our vaccine. So it's not anywhere near close to the whole virus. It's just a part of one protein. And that's one of the reasons subunit vaccines are safe. So there's no whole virus in there. It's not a inactivated vaccine where you have to worry about whether or not your vaccine is fully inactivated or anything like that. It's just one little part of the virus.
And so others are using this inactive part of a vaccine, part of a virus, I understand?
So that is one route to make a vaccine, and actually we have a lot of vaccines that are quite effective using that route for vaccination. We, sort of the global we, tend to not like that route with higher level pathogens. So SARS-CoV-2 is a level 3 pathogen. So inactivating it, even though you can improve that, generally that's not the route you go with higher pathogenic viruses that tend to be used for pathogens that are slightly more benign.
Let me step back if I could. Tell me what a level 1, a level 2, and a level 3 pathogen is.
Sure, a level 1 pathogen is something that's not even a pathogen. So that would be bacteria that are not really capable of infecting you, bacteriophage. So really something that can't even infect a human and is not going to cause any disease.
Okay.
Level 2 pathogens are something that have the potential to cause disease in humans, but generally would not under sort of lab conditions. So either it's very hard to get infected, you need a high dose, or the consequences of being infected aren't that grave.
Can you give me an example that people would know?
Sure, E. coli would be a level 2 pathogen
Okay.
or
Common cold.
sure.
Okay. And then level 3 would be SARS, MERS, and COVID-19?
Correct. Yeah, that would fall in a level 3. Anthrax would be a level 3 bacteria. So that's something where we don't have a good treatment. Getting infected is, presumably relatively easy, or it requires a lower dose, and the consequences are much greater, other than with death or severe illness being a possibility.
Just
And
out
then,
of
of course, the
interest,
top is level 4, which
just
we
out of
will
interest, what would a level 4 be?
So that would be something like Ebola. So that would be a virus that doesn't have a preventative, has high consequence, high lethality, easily transmissible.
Okay, and so let's go through the. You've described now some of the different ways of producing a vaccine. What's your success been in the past?
Here at VIDO, or you mean for other coronaviruses, or I kind of wonder at VIDO.
With your partial, what do you call it? A partial. You're using a part of the virus, just the spike on the top of it.
Right. So one of the reasons we're going ahead with this concept is coming out of data that we have from MERS coronavirus. So there we were intending to make a vaccine to use in camels, and we were also taking a subunit vaccine approach where we were taking this essentially the same portion of the spike protein, but for MERS coronavirus, combining that with our adjuvant. And in the case of what we were doing in our lab, we were using alpacas as a model for camels. And that vaccine worked surprisingly well. So it was preventing an upper respiratory tract infection in the alpacas. To be honest, actually, to a much greater degree than what I had expected with a subunit vaccine. And so that's something we had been working on. And so the same concept now is being used for SARS-CoV-2, where we're taking a subunit again with the same adjuvant and using that as our vaccine.
What do you think about your prospects versus any of the others? Have you taken a look at the Moderna, the Oxford University, University of Waterloo strategies, or do you just work on your own?
Well, you really need to pick what you're going to focus on. So we are focused on our own strategy. Of course, we pay attention to what other people are doing and their potential successes or failures also. For coronaviruses, there is no approved vaccine for humans for any coronavirus. So that is part of the problem. We don't have something to build off of, where we know sort of the best route to make a vaccine for a coronavirus, for a respiratory coronavirus for humans. And so I think that's why you're seeing all these approaches being done at once. Moderna's with their RNA approach, us with a subunit approach, others with an inactivated vaccine approach, other groups doing DNA-based vaccine approaches.
Why is there not a vaccine for any coronavirus? Is there something more challenging about a coronavirus?
So I think there are some challenges, but I think a major driving factor has not been, depends how you think about it, there has not been a real high requirement for one. So a lot of the coronaviruses that affect humans in a respiratory way cause essentially a common cold, although not for everyone. Some people do have more severe disease, but most people would have a common cold. And so we generally don't bother making vaccines for viruses that cause sort of minor illnesses that aren't causing severe morbidity or mortality. Of course, we had SARS-CoV, like the first SARS, back in 2002.
And you were working on a vaccine for that, were you not?
So I was not, but VIDO was at one time. I wasn't there yet. I was still a master's student during SARS. What happened there was SARS came, there was an outbreak, sort of got distributed around worldwide, and then it went away, and there were no more cases before there was even time to develop or really test a vaccine back then. So there were no other SARS cases. So the vaccine concepts that were developed for SARS were never ever tested in humans, or at least not thoroughly. And so that you never had a concept vaccine that actually proved to work for SARS. And SARS has never come back.
Why did SARS go away? Is it because it wasn't nearly as infectious as COVID-19? Did we do a better job with social distancing then?
So I don't think so. I think it has to do with transmission. It was not as transmissible as SARS-CoV-2. Why that is, I don't think we know. We still saw worldwide spread quite rapidly, but we were lucky and it kind of contained itself to hospitals and, for the most part, hospitals and elderly care homes were sort of hardest hit outside of the initial region in China. And I think you didn't have that much spread into the general public. And so that's when we were also able to get rid of the source. So civet cats were identified as the likely source. Those were removed from markets and culled. So we stopped transmission into human populations and we stopped the human-to-human transmission. And we're able to do that, and that was sufficient to control it. Obviously with SARS-2, we haven't been able to do that. In between the two, we've had MERS coronavirus, which continues to circulate in the Middle East. Which causes much higher lethality. So about 1 in 3 people infected with MERS end up dying still. Vaccines there move slowly because there's still less than 3,000 cases or so of MERS worldwide since 2012.
So it's far less infectious.
Possibly, yes. Transmission seems to be difficult. Chains of transmission don't go on for long. So there you get camel to human transmission and some limited human to human transmission, but always just in close contacts, like family members in the household or again in the hospital. And once those are controlled, that's sort of the end of your outbreak. Clearly, SARS-2 is much easier to transmit in the community.
Though from what I understand, the data coming out of China is that 80% of the people that got infected at the home.
So that makes sense. Again, you have your close contacts, longer periods of time, closer contacts. That's likely where transmission occurs. The highest percentage or the highest efficiency, but clearly it's happening in the community as well. Otherwise, you would be able to just sort of quarantine individual houses, and then you should be able to control your outbreak.
Yeah,
and this clearly hasn't happened, right?
But that is to a certain extent what they did in China. Is they either quarantine houses or they quarantine people in houses away from the balance of the house. And we certainly haven't done that in Canada and the United States. Off topic just for a second here, but the WHO a week ago on the Monday came out with a report that said that asymptomatic transmission was very rare. And then on the Tuesday, and it was in a quote, walked back that position. Are you familiar with that dispute at all?
I'm familiar with them walking it back, yes. That's one of those things that's very hard to prove because asymptomatic people generally wouldn't even know they're infected unless they just get randomly tested. And then you don't really know exactly what's going on. I'm sure asymptomatic transmission from the data seems to occur. I think nobody still knows how big of a part or how big of a role that continues to play, whether or not it's very important, of sort of like intermediate importance, or of low importance. It's one of those very hard to follow things because those people aren't picked up rapidly unless you do widespread testing all the time.
I was shocked that I think it was the chief scientist at the WHO said one thing on the Monday and then backtracked on the Tuesday.
Well, science is corrective. That's why we do that. And I think that's a responsibility that lays on us. So if we say something wrong one day, we might correct ourselves the next day. And that's how science works. And usually that occurs over a much slower period of time. So normally you publish something and then you realize it's wrong and correct that later, or that someone else corrects it for you. But I would say the public may criticize that as people don't know what they're doing, but I think it's important for people to admit when things need to be corrected, much more so than just sticking to your guns, right?
Yeah, well, I agree with you. If you're wrong, then you got to admit that you're wrong. But it's almost sounded to me like it was a political or expedient thing to backtrack because they wanted to get people concerned about wearing masks and social distancing and all that kind of stuff, rather than something that was actually rooted in science, particularly if you hear this Chinese, there's another report that came out that said that it was almost impossible to get it out of doors. So the vast majority of people got it in family units and got it indoors. And all this wearing of masks and social distancing, physical distancing would go out the window if all of a sudden it was proven that it was very difficult to almost impossible to get it out of doors when people have tried to prove that it can spread, the droplets can spread. Anyway, that's a whole different thing. We are having, I think, an interesting conversation with three very smart gentlemen from the University of Saskatchewan from a company that is working on a vaccine. The company is a 20-year experience. Is that correct? 20 years in vaccine development. It's called VIDO INTER Vac. And we're going to take a short break and come back more with these gentlemen in just a minute. Stay with us. Well, welcome back to the Brian Crombie Radio Hour. We're chatting tonight with Darryl Falzarono, Volker Gerdts, and Paul Hodgson. They're all research scientists at a company called VIDO INTER Vac, which I thought was around for 20 years, but Paul corrected me during the break and said you've been around for 40 years. Is that correct, Paul?
45 years this September.
45 years, fantastic. And I've worked on numerous different vaccines in the past, and they're one of the leading companies for vaccine development in Canada, and the first vaccine in Canada to get into animals. I think that's true. Let me, if I could, ask either Volker or Darryl about phase 3 clinical trials. So my understanding is phase 1 clinical trial is tested in animals. A phase 2 is tested in healthy humans. No? Okay, please correct me.
No, so what phase 1 is in humans, and that's really safety testing. It involves a small number of volunteers, typically around 40 to 60, somewhere in that range. Usually starts with an age group of 18 to 55, somewhere in that range. And it's just to look for whether there's any unwanted events to the vaccine.
Okay.
Then phase 2 is then a follow-up with now a larger cohort. You again assess safety in more people, but you also then start to measure immune responses in these people. And the phase 3 then is really to take now your vaccine technology out into the field, into thousands of people, and see how well it does out there. So you continue to monitor the safety, you continue to measure the immune responses, but you're also looking at whether there's actually any reduction in disease incidence in these individuals and so on.
Okay.
So that's a challenge that all of us are facing right now with the decreasing numbers in some countries. There was some concern where to do these phase 3 trials. So that's something that will have to be coordinated over the next year. And that's where global organizations like the WHO or CEPI are playing a big role.
So tell me about the ethics of a phase 3 clinical trial. So what you're doing is you're testing efficacy in a large population. And so you've got to give it to people in the community, and you don't know whether it's you've now proven already that it's safe. So you know that you're not going to be infecting people, but
Correct.
Really what you need is for people to be exposed to the virus to see if they then get infected or not, to see if the vaccine works. Are you actively trying to get them exposed, or is it just by chance?
Yeah, it's just by chance, and that's why you have to have such high numbers. There is no active exposure. People are, and this is different, people are talking about potentially doing challenge of humans, so infecting people purposely with the virus. But that's a completely different story. A classical phase 3 trial is you vaccinate thousands of people and you see how they do out there. And so that's, for example, what is currently being done in the UK, where the Oxford vaccine is now being tested in phase 3 trials.
And when you say thousands, are we talking 10,000 or 2,000?
It depends on the study design and your groups and all that. But I think it's for the Oxford study right now, it's 8,000 people.
8,000 people. That's a lot of people.
It really depends on what the regulator wants and on the incidence rate of your vaccine, of your disease. So you have to have a control group and a vaccine group, right? And in both, you have to have enough individuals to be able to detect differences in the disease incidence in these groups. And so, of course, that will depend on what the overall disease rate in that country is, or in that region where you do your trial.
And the people have to randomly be selected into the one arm versus the other arm so that they don't know whether they have the vaccine or not. Is that not correct?
That's correct.
And so will people still be in that situation, you know, wearing a mask, socially distancing, trying to do those things to not get infected? Or will you be telling them, no, go and try to get infected because we need the numbers?
So that's all part of the study design, and that's how the regulators will regulate it for each individual vaccine and for each individual country. It all depends on how each of these trials is going forward. So there is no general answer.
I think that, this is Paul speaking, I think that's key, because one of the fears of this particular one, and we saw it with SARS, is if the virus actually goes down, you will never you'll never gain the statistical significance you have to satisfy the regulators, because the virus has disappeared on its own. So when the virus was dropping, when the virus was being reduced in China, they didn't know whether China would actually have enough clinical cases that the vaccine would actually show that it was effective and pass phase 3 clinical trials for a full regulatory license.
That's right.
But it seems like Latin America and South America and maybe even Russia are skyrocketing in their numbers, and so you may not be able to have those numbers in Canada, but you should be able to do tests elsewhere, should you not?
Yeah, and the UK has sufficient numbers right now. As I mentioned, the Oxford vaccine is going into phase 3 trials in the UK.
How did they get to phase 3 so quickly? Is that a regulatory, were they just faster than you guys, or are they getting more lax regulatory review?
No, well, no. So they have a different technology. They're using what is called a vector. And so they already have a platform that they had tested for other vaccines, including TB, malaria, HIV, and so on. And so there is more safety information on the platform itself available. However, we don't know whether the technology works or not. It's a newer technology. It's not as proven as ours. In fact, there's at the moment no adeno-based vaccine out there in humans. The same with the RNA vaccine from Moderna. So these are new technologies that haven't proven yet, haven't demonstrated that they actually work other than in animals. And so that's the risk there. But because they have been so extensively tested at the moment, there is more safety information available on them, specifically on these platforms that they have. Our approach is slightly different in that we're not using a platform. As Darryl described, we're using the spike protein, a part of the virus. And for that, you have to go through a full regulatory process, including full safety assessment. It takes a little bit longer, but the advantage of our technology is that it's proven that in principle it works, and then it's also easily scalable. So once we can start manufacturing, we can easily make millions of doses per run.
Paul, maybe we can talk a little bit about that. You say that you're responsible for business development, so I presume you're the gentleman that's gone out and got funding. I understand you've got money from the government of Canada, Saskatchewan, $4 million, government of Canada, $28 million, Dalhousie University of British Columbia, South Korea Institute of Vaccine Institute, and the Bill and Melinda Gates Foundation. That's a whole heck of a lot of good people to get in your funding group.
So we've been fortunate building on our history and have been very successful in gaining funding to drive not only our vaccine forward, but some of the manufacturing aspects at our organization. So yeah, it's been good, but it's, again, our mission is protecting Canada and the world from infectious diseases. So all of this is aimed at driving the ability to have a vaccine available for Canadians as soon as possible.
We hear, at least I do, on the U.S. news on a fairly frequent basis about how they're sort of spec manufacturing vaccines as they're still testing it. Are you doing the same thing?
So, as Volker alluded to, some of these have platforms, so they are companies, and they took a fairly big economic risk of developing this to something called GMP, which is required for human clinical trials, at the same time as they're trying to move forward through animal trials. So we actually demonstrated that our vaccine worked in animals, and now are producing the clinical grade material. So I don't think anyone at this point really knows who will actually have a vaccine available for the broader use sooner, but we know ours, at least in the ferret model, and we'll know soon in the hamster model, works. And these other companies don't have that data yet, or it's not as clear.
So when do you think you'll start manufacturing commercial quantities? During the phase 3, during the phase 2?
So we are planning to do it ASAP. We should have at least the initial clinical grade material very, very soon.
Excellent. Well, good luck on that. And what about this manufacturing facility? Where is it, and what's the status of its construction?
Sure. So this is something I've personally been working on for over a decade. The federal government had started to fund this facility a couple of years ago, but as part of this current pandemic, I think quickly realized that there was the potential for borders to shut down, and that we had to look at it from a national perspective. What do we have in Canada to be able to produce vaccines for Canadians? So that's when they brought the next tranche of funding, which should fully build it. It's contained in our level 3 facility. So it will be, to my knowledge, one of the only facilities in the world that will be able to produce vaccines in containment level 3. And from a flexibility of design, I don't think there's any other facility in the world that will meet that. So we hope to have that up and running within a year.
Excellent. And give me a sense of how big is it in square feet? And do you have to have these air pressured rooms and stuff like that for vaccine development?
So yes, we have to, it's part of the level 3, so it would have all the standard operational aspects of the level 3 facility, which is both positive and negative pressurized rooms. And from a capacity perspective, we'll have up to 1,000 liter scale. So that's fairly significant. One of the things everyone asks is, well, how many doses of vaccine can that produce? Unfortunately, that's a fluid answer because it depends on how efficient the production is. But we would anticipate, based on ours, that we will be able to produce in a year, probably, depends, but it's at least 20 to 100 million doses of vaccine of our vaccine.
20 to 100 million doses. So more than Canada needs.
Probably more than Canada needs, and that's part of another thing that's been raised by the World Health Organization and governments. Is it's not so much the developed. We know that infectious diseases disproportionately affect the developing world, and even this one of the big concerns was, as this moves forward, if it gets into the developing world, they don't have the infrastructure to deal with what could be the outcome. And in the developed world, we knew that we were talking about respirators and all the other key medical equipment that we didn't have enough here. So you can imagine, with dense populations, how the developing world would have challenges there. So infectious diseases are called infectious diseases because they spread, and a lot of these poor nations have very, very dense populations in their cities.
Well, that's impressive that you're building this facility. How many square feet is it?
I think it's about 5,000 or 6,000.
Okay. And it's located in Saskatchewan?
It's in the level 3 facility, yes.
At University of Saskatchewan?
Yes.
Excellent. Okay. And are you guys private or public, your company?
So it's not a company, it's a research center of the university.
So those of us that play in the stock market can't go and invest in your company and make tons of money on this vaccine?
Oh, not currently.
Not currently. Oh, so you're planning it. Okay. Well, when you want to do the IPO, come and see me.
We need a mechanism to get the vaccine to the marketplace, and big pharma is obviously one of these phase 3 clinical trials are very, very expensive. So big pharma are one of the groups that have the ability to fund that. But as you saw on the US side, and we'll see if it happens on the Canadian side, the US government put huge amounts of money to companies to ensure that they had the vaccine produced in the US and available for US citizens. And the Canadian government has also done similar things.
This sort of nationalism attitude to both PPE and vaccines and everything, is this the first time that we've seen this, or is this something that I didn't realize for SARS and MERS and other things? You know, you think about the 2009 financial collapse, the whole world got together to come up with a coordinated plan, but it really seems like global supply chains are breaking down. PPE that was supposed to come from 3M to Canada got sent by Donald Trump to stay in the United States. And now you're saying that every country has to be almost on its own developing vaccines. What's happened?
So I don't want to leave that impression that every country has to be on its own. But as Darryl alluded to earlier, this virus right now is probably killing on the order of 3 to 4%. We work with another coronavirus that kills 33% of the people it infects. So can you imagine what would have happened to transborder travel, distribution chains, etc., if we had a virus that was this infectious, but instead of the current death rate, multiply that by almost 10 times, 8 to 10 times. Borders would have shut down, all of this. So don't think of it as nationalistic like that. It's think of it in an emergency situation. What do we have when the entire world could potentially shut down? What do we have to help our population? And it's not the first time it's happened for sure. We know that in '08, in the flu, that the US government tried to procure as much of the flu vaccine as they possibly could, and controlled a large percentage of the world's flu vaccine at that time. So it's happened before, and leaders of countries will watch out for their populations, and the scientists and nonprofits like us that we try and collaborate worldwide. And we're part of regular 2 to 3 times weekly calls with the World Health Organization, where people are sharing data and trying to solve worldwide this problem by collaborating as best as we can.
Great answer, Paul. We're going to take a break and come back with some concluding comments from 3 gentlemen at the University of Saskatchewan with this company called VIDO INTER Vac that's working on a vaccine. Stay with us. Well, welcome back to the Brian Crombie Radio Hour, Saga 960. We're chatting with 3 gentlemen from the University of Saskatchewan that work with VIDO INTER Vac on a vaccine development for COVID-19. Volker, maybe I can ask you, what have you learned here that you can apply to potential viruses, pandemics in the future? You know, a lot of us have probably seen this Bill Gates TED Talk that he did a couple years ago that said that this is coming. And then there was a movie that really predicted what was coming. And then I had the former health minister of Ontario, Tony Clement, and he said that we did a whole royal commission on SARS and came up with a whole bunch of predictions of what we needed to do, recommendations, and we didn't listen to them. So what are we learning now? What are we doing now in case something comes up again in the future?
Well, so I think we're learning, number one, that pandemics will happen. The next one is just around the corner. We've seen now 3 coronaviruses in 18 years, but there were flu viruses and so on. So it's just a matter of time before the next one arrives. Number 2, I think we're learning, or we're seeing, or we're all experiencing that our current strategy of developing vaccines is simply not working. It's super fast the way we do it at the moment, but it's still really slow when you think about all the people that are dying right now and the trillions of dollars that global economies are losing at the moment. So what we're focusing on here at VIDO INTER Vac, and that's really our mandate for the future, is to be proactively addressing these pandemics, trying to predict what the next pathogen might be, what it might look like, develop vaccines that would be available and stockpiled before the outbreak even occurs. So that, of course, sounds very futuristic and blue sky, but the science nowadays allows us to look for some of these patterns, and based on these patterns, we might be able to predict or what the next pathogen might look like, and then that will allow us to develop a vaccine for it. So it's really a combination of structural science, modern advancements in science, artificial intelligence, computational bioinformatics. Everything is coming together to now predict what evolution might do over the next few years.
Now, a lot of us have probably heard these rumors about there being a WHO-sponsored lab in Wuhan that was actually developing a bunch of pathogens to do exactly what you're talking about, find vaccines for them. Isn't there a risk that as you think about what's in the future, you actually develop it?
So what we're looking for is we're trying to understand what the similarities of these viruses are and what the regions are that we might want to attack with a vaccine. So it's not that we are creating a zombie monster virus that the next time around will kill everyone. What we're looking for right now is patterns, structures in these viruses that are similar and that we can target with our vaccines.
Darryl, you said earlier, maybe it was Volker, I can't remember. One of you said earlier that with SARS, we stopped the selling of a certain cat in markets. I understand this virus may have come from bats. Is there a similar effort to stop the selling of bats in public markets in China?
So we don't really, likely an ancestor of this virus came from bats. Whether or not this virus actually came directly from bats or through some sort of intermediate host first, that still remains to be determined. There's certainly been a little bit of movement in restricting some of the wild animal markets, I think, in China. But that's sort of an entrenched activity, so I don't see a total scale back of that happening anytime soon. With SARS, it was easy. It was civet cats. Those aren't a huge proportion of animals being sold, so they were removed from markets, and ones that were around were culled. So that doesn't have huge economic impacts. Shutting down that whole system likely would.
But, you know,
so I doubt that happens.
have,
and the other thing is trade of bats for consumption is not super common.
didn't we have foot and mouth disease or something like that, where we got mad cow disease, where we got rid of cows throughout the UK?
Absolutely, yes. And that's, you have to weigh the pros and cons of doing that. In that case, that was the only way to eliminate them, you know, mad cow and TSEs in humans. Likely there's less.
But we didn't really get rid of cows in the UK.
And that too, yes. It wasn't all the cattle. Yeah.
Okay. Darryl, it sounds like you know a lot about this virus. Let me ask you one question, if I could, completely off the vaccine topic, but it's been something I've been perplexed with. I've heard that 80% of the people that get put on a respirator still perish. Why is this virus so tough to treat? Why would these respirators not work for 80% of the people?
Well, not that I'm a physician, but the respirator is only part of it, right? It moves air in and out of your lungs when you're incapable of doing that. But if your lungs are not capable of exchanging oxygen efficiently to keep you alive, it doesn't really matter what the respirator is doing. So the people who don't survive have lungs that have extensive enough damage that this doesn't get them through it. So that's only
get on the respirator.
The respirator is not a treatment, right? It's only a supportive measure. So you need to do something to either block the damage to their lungs before that happens, or to allow it to repair over time. And at some point, if your lungs are just not functional, it doesn't matter what you do.
Now, you mentioned AIDS. I think it was Volker that mentioned HIV/AIDS earlier. We don't have and never have developed a vaccine for HIV/AIDS, but we have got treatments. Are people like you, as smart as the three of you, working on treatments for coronavirus?
Yeah, so like we said earlier on, our organization here is really, as a national facility, is involved in a lot of contract research. We're working with other companies to test their drugs, their antivirals, their therapeutic approaches. So we're not only working on our own vaccine, but we're also helping others in developing their technologies.
Volker, maybe I can ask you a last question. You mentioned a challenge trial earlier. I interviewed a gentleman from Mount Sinai that he had proposed a trial where people would get infected with the virus, volunteer to get infected with the virus in a healthy, healthcare-oriented setting so that they would get treated, but to create effectively herd immunity. Does that make any sense to you?
So I think that will be very risky if you purposely try to infect everyone to bring herd immunity up to above 80%. You are also infecting then a lot of people that might be very susceptible to the disease. So I don't think that's a good idea.
When do you think we're going to be rid of COVID-19? When are we going to get our lives back?
When we have a vaccine available for it.
So it's only a vaccine that's the solution, and that's what, a year away?
Well, I think, you know, as you alluded to in your comment, so antivirals and drugs are very, very potent, and they for sure will help getting patients out of hospitals and allowing us to control the infections, or, you know, severe cases, and that will reduce the shedding and all those things. So transmission will slow down, but effectively to control an infectious disease like this, it's when you reach a level of herd immunity through a vaccine that stops transmission of the virus. Well,
Volker Gerdts, Darryl, Falzarono, and Paul Hodgson, all of VIDO INTER Vac, a 45-year-old company doing vaccine development at the University of Saskatchewan. Thank you so much for joining me tonight on the Brian Crombie Radio Hour. I really appreciate what you guys are doing, and Godspeed, because we need your vaccine.
Thanks for having us.
Very much.
Thank you. Good night, everybody. Thanks for joining us.